Hello everybody and welcome back to the Sanctuary. I'm your host, Professor C, and we're going to wrap up the recent lectures in A&P we've done with a self-test. This will be test yourself number 8, and we will cover specifically muscle physiology that we've covered in the last few lectures. So let's give it a try. Question 1. What is the role of tropomyosin in skeletal muscle? Well, if we could draw a little actin filament like that, and we could put some binding sites for the myosin heads like that, the tropomyosin, remember, was kind of a ribbon that went around these and blocked these. Even though my picture is not beautiful, it blocked the binding sites. So let's see if we can see something similar to that. It is the contractile unit. No, it is not. That's a sarcomere. It wraps and protects each fiber. Now, that's endomysium is the term for that. It's an acetylcholine receptor. No, it's not that either. It has nothing to do with motor end plates. It's a regulatory protein. Yes, it is. That's an unofficial way to say that it hides other structures and can be moved to show them. And we are correct. Next question says, which of the following would be recruited later as the strength of a contraction increases? Okay, remember, recruitment tells us it's going to recruit smaller motor units first, and then as we need more and more, we will recruit the larger and larger motor units. So let's check out our choices. Okay, smallest units would not be later. They would be first. Motor units with the longest fibers. All right, longest fibers has nothing to do with it. It's the number of fibers, right? It's the number of motor units. So motor units with the longest neurons, no, that has nothing to do with it either. The largest motor unit, there you go, the one that has the connections to the most number of fibers. So D is our answer. Which of the following structures functions in calcium storage? Okay, I'm thinking immediately of sarcoplasmic reticulum and the cisternae. Let's see. Mitochondria, no, they do not do that. Sarcoplastic reticulum, that looks like a really good choice for the answer. Epimysium, no, that is a layer of connective tissue wrapping around a muscle. And transverse tubules don't have calcium in them, although they do carry action potentials down to the SR to release that calcium. So B is our choice. Which of the following structures are composed of only myosin? All right, let's check it out. Thin filaments are actin, so that's not it. Z discs are found right in the middle of that light band where we find actin and titin, so that's not it either. Thick filaments are made of only myosin, so that looks like a really good choice for our correct answer. D is sarcomeres. That is wrong, so C is our best one. Only one. Rigor mortis occurs because blank. At the end of the day, it's because we've run out of ATP. That's the answer. So let's find it. Actin and myosin have broken down? Nope, not yet. They will eventually, but not yet. The muscle cells are dead. That's not the reason why. Potassium leaks from the dying cells. That's not correct either. There it is, ATP. It's got to be about ATP. No ATP is available to uncock the myosin heads. Okay, next question is, muscle tissue has all of the following properties except extensibility. Well, yes, it is extensible. Contractility is contractile or contractible. Does not secrete, so that's probably going to be our winner. And it definitely is excitable, so C is our answer. After nervous stimulation ceases, what prevents ACH in the neuromuscular junction from continuing to stimulate the muscles? So the question is talking about if there is a terminal knob and it is secreted acetylcholine and it's moving across to the other side where there are receptors waiting, what is preventing it, the acetylcholine, from continuing to stimulate the muscle? Well, there's a couple of mechanisms we talked about, but it probably has something to do here with an enzyme will degrade it. So let's look. Calcium efflux, no, that is not right. Tropomyosin has nothing to do with this situation. Acetylcholine esterase, there it is. There's the enzyme that destroys acetylcholine. That is a wonderful choice. also known as ACH, ACE. And troponin, nope, that's not it either. So C is our winner. What type of muscle cells are uninucleate, have no sarcomeres, and almost no gap junctions? Okay, this is a little trickier. Uninucleate immediately tells me it's smooth muscle. But the thing at the end there, well, no sarcomeres reinforces that it's smooth muscle. But the almost no gap junctions, it's kind of making me choose between single unit and multi-unit smooth muscle. So almost no gap junctions would be the multi-unit, right? So let's look. Single unit, multi-unit, skeletal is definitely wrong. Cardiac is definitely wrong. All right, B is it. Remember, the single unit smooth muscle is myogenic, and it generates its own signals. Muscle tone is A, a partial sustained contraction. Well, that's a perfect definition, so that looks like going to be our winner right there. Tightness of muscles after exercise, no. The inability to contract, no, that's fatigue. And a result of training and conditioning, that's not true either. You can have toned muscles without training or conditioning. So that is correct, A. The cell membrane of a muscle fiber is called A, and this is just a definition. The term is sarcolemma, so let's find it. Nope, not sarcoplasmic reticulum, not sarcomere, that's the contractile unit. There's sarcolemma, that's going to be our winner. And not a sarcomesium, that's a made-up word. C is the winner. During an isotonic contraction, blank. Well, muscle always shorten. Always is a key to watch out for, right? Muscles always lengthen. That's kind of dangerous too. A load is moved. Tension decreases. Well, that doesn't make sense either. So the two always is suspicious, and the decreasing tension is suspicious. But if you remember, our definition of isotonic means we're going to actually overcome the resistance of the load and move it through space. And that is our answer. Which type of connective tissue wraps individual cells? Well, epimesium wraps a muscle, perimesium wraps the fascicle, and endomesium wraps the individual cells. So that's what we're looking for. Nope, epi, peri, endo, there it is. And myomesium is a false term, it's fake. So endomesium is our winner. Multi-unit smooth muscles are controlled by autonomic neurons. Remember, they're diffusely branched and they have lumps in them, bulges in them, and they're called varicosities. So let's look for the term. Cisternano. There it is, varicosities. That should be our winner. And interneurons and transverse tubules are bogus. What produces the striations of a skeletal muscle fiber? Well, it's going to be that alternating A band, I band, A band, I band. But let's see what they're talking about here. The overlapping arrangement of microfilaments. Well, that is what we're talking about. Actin overlapping with myosin and producing the A band and I band. That might be our winner. Sarcoplasmic reticulum. No, that's not correct. Pigments within the sarcolemma. That's very, very incorrect. And elastic connective tissue is wrong also. So we got 3 horrible choices and 1 decent one up top. So that's our winner. The most immediate source for replenishing ATP in a muscle fiber. Okay, there were 3 mechanisms we talked about. We talked about an anaerobic way to do it, an aerobic way to do it. But the most immediate source on any test you ever see is creatine phosphate. So let's just find our answer. Glycogen, no. Oxygen, no. Protein, no. Creatine phosphate, that's our winner. Within seconds, we can use that and turn it into ATP. What is the role of calcium in the terminal knobs of motor neurons? Okay, remember, calcium will come into the terminal knob when the action potentials run into it. And that will activate a chain of proteins called synaptotagmins. And they will put acetylcholine into a vesicle. So calcium directly activates synaptotagmin. So let's look for that choice first. It's used to cock to myosin heads. No, that's ATP does that. It's used to activate synaptotagmins. That's our winner right there. It's got to be. It's used to synthesize ACH. No, it is not. It's used to generate action potentials. No, it is not. So we got it right, B. The contractile unit of skeletal muscle is this is by definition called a sarcomere. Sarcomere, let's just find it. Sarcolemma, no, that's the cell membrane. Sarcomere is the answer. Endomysium is what wraps each fiber. And actin is something to do with contracting within the contractile unit. So sarcomere is our winner. The time it takes to stimulate a fiber and elicit a contraction. Okay, all that time it takes to do this, and finally get the actin and myosin to slide over each other. That's called a latent period. So there it is, first thing, latent period. Let's make sure. Recovery period, nope, that's for exercise. Refractory period is a good choice, but it turns out to be not the answer. And EC coupling is not the answer either. So, latent period. During anaerobic metabolism, okay, pyruvate is converted to, well, it's converted to lactate or lactic acid, if you prefer to play that game. So glucose, nope. Ultimately it will be, but immediately it's turned into lactic acid. There it is, choice D on our list. What is the functional role of transverse tubules? I gave this one away already, but they will take action potentials deep into the tissue and tell the cisternae to dump out calcium. So regulation of calcium, ooh, that's not the best choice, but maybe it is. Let's see what else we have here. Bringing APs deep into fibers, that's a much better answer, even though A is arguable, that it is going to be regulating calcium at some point, but technically, and that's the best way to be correct, right, is the technical way, that should be B. Connecting actin and myosin, no. Diffusion of ACH, no. So, okay, we were right. A is, I mean, I could make an argument for A, but B is better. B is best. Which of the following describes the cells of a single unit visceral muscle? All right, let's check it out. They are structurally separated from one another. No, they're not. They're myogenic, and they have lots of gap junctions, and they connect to each other electrically. They're neurogenic. No, they're not. That's the multi-unit smooth muscle that's neurogenic, relies on the autonomic nervous system. They, single unit, create spontaneous action potentials. That is true. They are myogenic, and that is the definition of it. They control the iris of the eye. No, that's multi-unit. So C should be the answer. Got it. What is the role of calcium, again, in the sliding filament theory? Okay, we saw calcium in the knob a moment ago. Now we have calcium here in the actual sliding filaments. Well, remember, calcium will bind to troponin, and this will allow actin to twist and pull tropomyosin off of the sites where the myosin heads will bind. That's a mouthful to say, but essentially it will bind to troponin. Let's see if we can find an answer that looks like that. Calcium binds with troponin to move tropomyosin. Perfect. Calcium binds with tropomyosin to move troponin. No, that's backwards. Calcium binds with myosin. No, it does not. Calcium is a major component of actin and myosin. No, it is not. So we started off with the winner right away. The time during which a muscle is unable to respond to additional stimuli is called blank. Now remember, we just went over this latent period idea. This is the refractory period under which you cannot stimulate a fiber that's been stimulated. So latent is not this one. Recovery is an exercise phenomenon. Refractory, that is our answer. And that is not a bogus excitation coupling. Don't even know what that means, to tell you the truth. And that's the end of that test. How'd you do? If you didn't do so hot, go back, check out some of the videos that explain all of those answers within the lectures. And thanks for watching it, of course. I will see you for more videos, and check out all the other ones I've got in the library if you want to learn more about AMP. See you for the next one. Bye-bye.